Okay, today's video is going to be on DNA replication. Replication, remember, means copy, to make another copy of something. So let's look at this and just do a little bit of review. Remember, we have here a double helix. It's twisted. This is a typical picture of a DNA molecule. And when that gets all wound up tight, it forms a chromosome. And in your cells, that's going to be contained in the nucleus. So this is what we're doing when we have our chromosomes. They look like this. And so now we want to figure out, well, how do we copy our DNA that's inside of our cells? And remember, we're doing that because we want to grow, repair, and replace things. So, by definition, DNA replication is the process of copying DNA to create a new cell. If you remember, the cell cycle, it starts with interphase. And in interphase, you have those three subparts, the G1 where the cell grows, and then the S phase where DNA copies itself, and then the G2 where there's more growth, and then there's mitosis and cytokinesis. Well, what exactly does it mean when we say DNA copies itself? So we're going to make sure that we talk about the way that we're making an exact copy. And that happens in 3 easy steps. The first step, DNA is going to break apart. So if you look at, here's a typical strand of DNA kind of unwound. And so what happens is some enzymes are going to come along and break the bonds between the nitrogen bases, and they split apart. The second step is we're going to have a bunch of nucleotides that are going to come along and attach to each side of our split apart DNA, and they're going to follow the base pairing rules, which remember, A goes with T and C goes with G. And then the third step, or the final step, is it's all completed and you end up with 2 identical strands of DNA. Each has an original side, the red, and then each has a new side. So where we started with 1 strand of DNA, now we have 2 strands. We split it apart and made new sides of the copies using the base pairing rules. So if you look at here, here's a strand of DNA with my nitrogen bases A and T, C and G, and so forth. It's going to split apart here. Some enzymes break the bonds between the A and the T, and then along comes some new nucleotides and makes a fresh new side. So A pairs with T, C with G, T with A with T, and C with G. So I have a completely new side. The opposite happens on the other side here as well. So I have 2 strands of DNA, each with an original, and then each with a new one. So the reason we do this is, again, so that we can pass on our DNA to the next cell, so that you can maybe use that cell to grow in size, to maybe eventually make a new individual, or maybe repair or damage cell that you have. So let's look at 1 strand of DNA following the nitrogenous base rules. If I have an A, a T, a G, a C, and an A, what would the order be of the complementary base pair? A would pair with T, T would pair with A, G would pair with C, C would pair with G, A with T. So I'd have to call over those nucleotides to make those bonds. Let's take a practice question here using our base pairing rules. In a double-stranded molecule of DNA, 10% of the nitrogenous bases are cytosine. What percentage are going to be guanine, and then what percentage would be thymine, and what percentage would be adenine? So let's look at and try and figure that problem out. Well, if 10% of the DNA is cytosine, then I know 10% is also guanine, because cytosine and guanine always pair up, the C and the G. So however how many C's I have to have the same amount of G's. So that means that 20%, because 10% and 10% is 20% of the DNA molecule is cytosine and guanine. Well, if I have 100% of a molecule minus 20%, that means I have 80% left over. So that means 80% of the DNA has to be made up of adenine and thymine. So there's 2 of them, so I divide the 80% into 2, so that means 40% of the molecule is adenine and 40% of the molecule is thymine, because however many A's I have to have the same amount of T's. And so that's how we can figure out complex problems and understand our DNA molecule. So why, again, does DNA copies itself? It's because all organisms, whether you're a complex organism like a human or a simple organism like a bacteria, have to reproduce, grow, repair, and replace cells. And remember, so here we were talking about mitosis. A zygote, a baby, goes through mitosis to create and grow new cells all the time, and it grows up into an adult. And eventually that adult has to produce special cells in our gametes so that they can come together so the process can continue on and on and on again. And that way we keep human beings on the planet, we keep plants, animals, and all organisms go through this process. And so DNA replication, or DNA copying itself, that happens in the S phase is very, very important for living things. These 3 videos, if you're still a little bit confused and want to watch some animations, are here for you to take a quick peek at. So this is when you're done. You're going to make some flashcards using your vocabulary. You can even do them on Google Read and Write if you want, or you can make it on a Quizlet. Then remember to do a learning activity, check your answers, and at this point now you're ready to take a quiz on the DNA molecule itself and DNA replication. Good luck. If you have any questions, come see me.